Hello everybody. Today we'll be learning something about cell, the basic unit of life. Now let us learn some facts about cell. Cells are the basic structural, functional and biological unit of all living organisms. Cells were first discovered by Robert Hooke in 1665 in cork cells. Now these cork cells are dead cells, but who observed them first? Robert Hooke in 1665, and with the help of a primitive microscope. It was not such an advanced microscope as we have today. Then in 1674, Anton van Leeuwenhoek first observed a living cell in pond water with the help of a self-modified microscope. Okay, now cells can be of different shapes and sizes. They can be rounds, spherical, elongated or spindle shaped, and they may vary in size from 1 micrometer in diameter to 100 micrometer in diameter. Okay, now organisms which have only one cell are known as unicellular organisms, and all those organisms which has many cells, more than one cells in fact, are known as multicellular organisms. So based on these, that is the number of cells that an organism is having, organisms are classified into unicellular organisms and multicellular organisms. Some examples of unicellular organisms are amoeba, chlamydomonas, paramecium and bacteria. And multicellular organisms are fungi, animal cells, plant cells, etc. Now here we see pictures of an animal cell on the left and plant cell on the right. We'll differentiate them and study in details about them later. The various types of cell in human body. You can see all of them: stem cells, fat cells, pancreatic cells, sex cells, skin cells, nerve cells, cancer cells, blood cells, and so on. Here we see a picture of a bone cell, a blood cell, a skin cell, sex cell, nerve cells, they're called neurons. Okay, now cells were first observed in 1665 by Robert Hooke. From then on, they have observed numerous cells, and we have now seen abundance of cells. But nobody was actually clear about what the functions of cells were. What do they do actually? So, Matthias Schleiden, a botanist from Germany, and Theodor Schwann, a zoologist from Britain, respectively in the years 1838 and 1839, and independently also put forward the cell theory, which rather illustrated what the main functions of a cell were. What did it tell? All organisms are composed of cells. Each cell arises from pre-existing cells. That is, if we see a cell today, that means it must have originated from another cell which existed earlier. And the third point tells us that every organism starts its life from a single cell. That means what? Be it a unicellular organism or a multicellular organism, its first stage in life was a single cell. That is what cell theory is telling us all about. Okay, now cells are broadly classified into prokaryotic cells and eukaryotic cells. We have the pictures adjacent to the names written here. What are prokaryotic cells? Prokaryotic cells are first of all primitive kind of cells. Okay, they are generally smaller in size, 1 to 10 micrometers, and they lack nuclear membrane. They also lack a proper nucleus, and the region of dense cytoplasm is known as nucleus. They also lack organelles like endoplasmic reticulum, mitochondria, Golgi apparatus, centrosomes. Okay, all these terms we'll learn later in the next video, in fact. Ribosomes are of 70S type, where S means sedimentation rate. Cell division is generally amitosis. DNA is circular and is found freely in the cytoplasm. Cell cycle is short, of nearly about 20 to 60 minutes. Cell wall is made of murine and microtubules are absent. So this is just a list of some features that a prokaryotic cell possesses. Next we'll learn about eukaryotic cells. These are the advanced cells, generally larger in size, 5 to 100 micrometer. They are well organized nucleus and is present with well developed nuclear membrane. The cell organelles are also well developed and the ribosomes are of 80S type. Cell division takes place through mitosis and meiosis. The DNA is linear in shape and found within the nucleus. Cell wall found in plants is made up of cellulose, hemicellulose and lignin, and microtubules forming cytoskeleton are present. Now let us just clarify the fact about prokaryotic cell and eukaryotic cell. What a prokaryotic cell is, we can compare it with a slum house. A room where there is a kitchen, a bedroom, guest room, study room, everything packed in a single place. Now you can compare that to a prokaryotic cell where all the functions are simultaneously taking place at a single place, and hence it is known as primitive cell. And in case of eukaryotic cell, you can obviously compare it with the recent bungalows where you have guest room, kitchen, drawing room, bedrooms, where different functions are performed in different areas or regions of the house. Similarly, in eukaryotic cells, different functions are performed in different organelles of the cell, and we'll learn about the cell organelles in the next video. So let us just move on. Okay, cell structure. So basically, the structure of a cell contains three parts: the cell membrane, the cytoplasm, and the nucleus. The cell membrane is the outermost covering of the cell. Cytoplasm is the jelly-like fluid that is within the cell membrane and outside the nucleus. And what is the nucleus? It is the central part of the cell. It is also known as the brain of the cells, which controls the entire functioning of the cell. The point to be noted is, in animal cells, cell membrane is there, but other than animal cells, certain cells also have a cell wall which envelops it in addition to the cell membrane. Is that clear? Okay. Now what a cell wall is? A cell wall is rigid, non-living but flexible covering of a cell wall which is present outside the cell membrane and provides structural support to the cell. So what is the first function of cell wall? Provide structural support to the cell. Okay. Next, it also protects the cell from outside attacks and infections. So what is the other function of cell wall? It protects the cell from outside attacks and infection. Next, it also helps tolerate much greater changes in the surrounding medium as compared to animal cells. It is particularly found in plants, fungi, bacteria, algae, and some archaea. Now, cell walls are made up of cellulose or chitin, but may vary depending upon the species. Okay, it is made up of three layers. It is made up of three layers: the primary cell wall, the secondary cell wall, and the outer cell wall, also called the middle lamella. Now, what the primary cell wall is? It is thin, flexible, and is generally found when the cell is growing. Okay. Secondary cell wall. So when is it formed? A secondary cell wall, when a cell completely grows and develops, then a thick cell wall is formed, which is known as the secondary cell wall. And the outer cell wall or the middle lamella is nothing but the outer covering. Okay, and it helps in adjoining two different cell walls. So it acts as a glue between two cells or between two cell walls of two adjoining cells. Okay, let us just move on. We'll now study cell membrane, and mind you, this is the most important part. It is the outermost covering of a cell, separating the inner content of the cell from its external environment. It is also known as the plasma membrane. It is made up of lipids. Major components is phospholipid. So what is the major component? Phospholipid and proteins, and hence it is also called the phospholipid bilayer. It is also called the phospholipid bilayer. It is a thin, semi-permeable, double layered, mind you, cell membrane is a double layered membrane which allows selective entry or exit of substances into and from the cell, and hence also called the semi-permeable membrane. So a cell membrane is known by different names. First of all, plasma membrane, a phospholipid bilayer, and a semi-permeable membrane. So what are the functions of a cell membrane? The major functions of cell membrane is to provide support to the cell body by providing support to it. The flexible nature of the cell membrane allows the cell to engulf food and other substances from its surrounding environment, and this process is known as endocytosis. So what is endocytosis? The process of engulfing food into itself or into a cell. It also performs functions such as cell adhesion, sticking to some cells, ion conductivity, movement of ions into and out from the cell, and cell signaling. It also serves as the base of attachment of cytoskeleton in some organisms and the cell wall in others. Okay, here we see some pictures of cell membrane, and this is the picture of phospholipid molecule, where we see hydrophobic tails and hydrophilic heads. So the round heads are hydrophilic in nature, that is, they are water loving. So from the picture we can interpret that both the exposed surfaces of the cell membrane, that is the inner part and the outer part, the extracellular fluid and the intracellular fluid, are in touch with the hydrophilic head, and the interior or the middle region of the cell membrane is made up of hydrophobic tail. Understood? Okay. Next main part of the cell is nucleus. So Robert Brown back in 1831 discovered nucleus. It is an extremely important component of a living cell and is generally spherical or elliptical in shape and is located in the center of the cell. Roughly at the center of the cell, not exactly at the center, but roughly at the center. It acts as the major controlling center of the cell and also called the brain of the cell. It contains the basic hereditary unit of the cell and carries it forward to the offsprings. Now, what are the major components of a nucleus? The nuclear membrane, nucleoplasm, nucleolus, and chromosomes. The nuclear membrane. So what is a nuclear membrane? Similar to a cell membrane, it separates the nucleus from the cytoplasm by a membrane, and that membrane is known as nuclear membrane. It is also double-layered structure made up of proteins and fats, which encloses the components of a nucleus. It has pores, which allows the entry and exit of nucleic acid and proteins. Okay, now within the nucleus there are several processes going on, and from there nucleic acids and proteins keep on moving out and in from the nucleus. Okay, a fluid-filled space or perinuclear space is present between the two membranes of the nuclear membrane. So between the two nuclear membrane, a perinuclear space is present. What is nucleoplasm? It is the fluid found inside the nuclear membrane. It is a little acidic and is also known as the karyoplasm or nuclear sap. So what is it also known as? Karyoplasm or nuclear sap. It is composed of nucleolus and chromatin. Okay, and it has an abundance of proteins, phosphorus, and nucleic acid. So nucleolus has an abundance of proteins, phosphorus, and nucleic acid. Nucleolus. It is a spherical structure present in the nucleoplasm and is not a membrane-bound structure. The main function of the nucleolus is to synthesize rRNA and to store genetic information in the DNA. Next, we'll be studying about chromosomes. In fact, the basic unit of hereditary information. Not basic, but the most important. These are thread-like structures found in the nucleus as chromatin materials, as we can see from the picture. It becomes visible only during cell division. It was named by Valdeir in 1888 and is capable of self-replication. It transmits coded information from one generation to the other and is also called as hereditary vehicle. We can just look at the image here. Okay. Now the chromosomes are divided into two parts, the P and the Q arm, with a constriction point known as the centromere. So this region where the two arms meet is known as the centromere. Okay, now a centromere can be located at different points. So based on the position of centromere, chromosomes are of 4 kinds: metacentric, submetacentric, acrocentric, and telocentric. Metacentric chromosomes are what? Metacentric chromosomes are those chromosomes where the centromere is exactly in between the P arm and the Q arm. Submetacentric chromosomes, the centromere is slightly off center, leading to a shorter P arm and a larger Q arm. Acrocentric chromosomes are those where the centromere is severely offset from its center, leading to much shorter P arm and a longer Q arm. Telocentric chromosomes, the centromere is found at the end of the chromosome, meaning no P arm exists. That is, these chromosomes, telocentric chromosomes, are not found in human beings. Okay, next. Okay, here we have just prepared a chart where different organisms and the number of pair of chromosomes that they are having is listed. So pigeons are having 40 pair of chromosomes, chimpanzee 24, cats are having 19, onion 8, dog 39, human beings 23, frog 13, pea 7, horse 32, tomato 12, and wheat 21 set of chromosomes. Genes and DNA. Genes are made up of DNA. Now what do you mean by DNA? Deoxyribonucleic acid, okay, which are carriers of genetic information from generation to generation. In some viruses, RNA is the carrier of genetic information, like retroviruses. Now you must be getting confused now. Previously I said chromosomes are carriers of hereditary information. Now here it is mentioned DNA. Let me be very specific in mentioning DNAs are long strands, double helical structures, and they are winded up so tightly to form chromatin fibers. These chromatin fibers during cell division get condensed and we see them as chromosomes. Understood? And these small segments of DNA are known as genes. Specific segments of DNA in the chromosome are known as genes. So chromosomes are made up of DNA and proteins, and these proteins are known as histone proteins. So who discovered or who proposed the structure of DNA for the very first time? It was Watson and Crick. And the structure of DNA happens to be a double helical structure joined together by nitrogenous bases. Friedrich Mischer was the first to isolate DNA from the nucleus of a pus cell. In small amounts, DNA is also found in the mitochondria and chloroplasts. Now these two are cell organelles which we'll study later. Okay, so this is the end of part 1. We'll study about the different cell organelles in the next part of this video. Thank you for watching and do like and subscribe.